Hi, my name is Chris Bennett. I'm an astrochemist from the Georgia Institute of Technology, and I'm going to be providing the critical eye from a scientist for the talk by Robert McFarlane on the origins of life, abiogenesis, and panspermia. So first of all, Robert, it was a very entertaining talk. Thank you. I enjoyed it a lot. I did notice a few gaps that I will try to fill in very briefly, and this is a very big topic. You give a very, very long presentation on this, but I'm going to try and do this in about 10 minutes or so, hopefully. So firstly, let's just give a quick definition of abiogenesis and panspermia. So abiogenesis is basically the idea that when the Earth began, there was no, sugars or amino acids, and all of these things had been made on Earth under processes that were occurring on the Earth's surface, such as by lightning, etc. Whereas panspermia entails that things would have been brought to the Earth that could have kick-started life a lot earlier. So this might have been just things like amino acids and sugars, or it could have been something as complicated as DNA. The definition of panspermia is a little bit iffy in that regards. So what do we know about the origin of life? And how early did it begin? So microfossils have been recovered in two locations where there are old rock formations still exist. These are in South Africa and Western Australia. And a lot of these fossils are found in rocks that are aged about 3.5 billion years old. There's more evidence for life earlier than this still. There's evidence of these porphyrin-like structures, which are very similar to the kind that you find in chlorophylls and other CO2 fixing pigments. So these are identified in sediments about 3.8 billion years ago. And there are also evidence in keratin shales. These are kind of like organic shales that are found in Greenland about 3.85 billion years ago. And these are depleted in 12 carbon versus 13 carbon by approximately 70%. Now this could be indicative of life, because today there's an enzyme that's responsible for CO2 fixing called rubisco. And this is actually a very selective enzyme, so much so that it can actually preferentially takes 12 CO2 out of the atmosphere versus 13 CO2. So as a result of this, all living things are depleted in 12 carbon versus 13 carbon. However, we know that prior to around about 4.1 billion years ago, there's a lack of rocks, and also from the cratering of ages of the craters in the moon, we know that there was this late heavy bombardment period where there's a lot of comets and asteroids that hit the Earth, and the frequency and size of these impacts was enough that they're thought to have been able to sterilize the Earth and volatilize an ocean prior to about 4.1 billion years ago. So on the Earth, there are many ways of making biological molecules, but how efficiently can they do so on the time scale between 4.1 to 3 billion years ago, where life would not have existed, to 3.85 billion years ago, where life may have already been flourishing. So though it sounds like a lot, 200 million years is actually not a lot in these timescales. So we have three different scenarios I've outlined here. The first is lightning, which is made famous by the Miller-Urey experiments. I think they're in 1953 or so. And here you have an atmosphere. You might have some molecules like CH4, water, etc. And you end up, the lightning will break them apart and then reform more complicated molecules. And this has been shown to make some things like amino acids to some degree. And this one in the middle is a hydrothermal vent. So they found in the bottom of the sea floor a lot of these sort of black smokers, they call them. And these can reach temperatures of about 350 to 400 Celsius, and can also be sources of a lot of organic chemicals. On the right here we have a volcano, which again can induce a lot of chemistry. Okay, so there's also the possibility that other than those three processes, the Miller-Urey synthesis, the volcanic outgassing, and hydrothermal vents, you can also get this exogenous delivery to the Earth via things like comets, interplanetary dust particles, which are basically the parts that come from comets. Usually when you see a meteor shower, it's due to a comet that's passed by recently and they're passing through that area. But also meteorites and asteroids. So all of these things would have been made as our solar system was forming. And you basically like go backwards, so the solar system would have come from like a pre-stellar disk, and this itself would have come from a dense cloud, and this would have come from the diffuse interstellar medium, where there are a number of complicated molecules, possibly even amino acids. So in this scenario, you have like a hot Earth surface, and you have these interplanetary dust particles that have come from comets, or even comets that are impacting the Earth to some degree and bring in a lot of organic molecules to the Earth's surface. So over around about 200 million years, it's predicted that this could have brought about 6 times 10 to the 20 grams of organic matter to the Earth's surface. That's a very hard number to get your head around, but to put that in context, that's around enough to cover the entire Earth's surface in a layer about 40 meters thick. This is also around about 50 times more than all of these other terrestrial sources combined. And there was a recent paper, I think in Science or Nature, that was explaining the isotopic ratios are consistent with the fact that about 50% of our oceans could be consistent with the fact that the water would have been formed in the interstellar medium. So it's somewhat plausible. I'm just going to briefly talk about panspermia being an option. So the question being, how complicated can molecules that are delivered to the Earth be? And in Murchison meteorite, they were able to identify a lot of amino acids. There were some things that looked a little bit like organs, but there was no life discovered there. So the question about panspermia is, could life have been brought to the Earth? There are ways that if you have a giant impact, you can actually transfer life from one planet to another, because rocks will get picked up from one planet during a giant impact and brought to another. So this could have happened that like perhaps life might have started on Mars, and then this rocks would have been brought from Mars to Earth, and then that's, then life evolved on Earth after it evolved on Mars. Similarly, this could have happened about 65 million years ago, when a giant impact caused the extinction of the dinosaurs. It may well be possible that this could have brought dinosaurs to Mars. Perhaps, maybe not. So the question is like, how easy is it for life to get transported between one body and another? And the thought is that it's very, very difficult, because the conditions in space are very harsh. There are a few life forms that have been shown to be able to do this, however. This cute little fellow over here is called a tardigrade, or for obvious reasons, a water bear. And these are very resilient little creatures. They've been shown to be able to like withstand about several hundred times the dose of radiation that we can. They can withstand temperatures from like almost absolute zero to temperatures above boiling. They can put up with incredibly high pressures, and also almost a complete vacuum. They can also go for food within 10 years and survive with a body weight of about 3%, body condition of about 3% water. So these guys might be able to make it on a journey within the solar system. Another candidate is Bacillus subtilis, and this was suggested by, I think, Francis Crick. So these are actually very common bacteria. They're found within our human guts. And the small thing you can see here is an endospore. And this is part of their natural life cycle. So they can go in, they can generate these spores when their environment doesn't suit them very well. So conditional stress, but if there's lack of nutrients, for example, they can just go into the spore cycle until the conditions become more favorable, and then they can come out. And these spores are extremely hardy. They can survive extreme conditions such as drought, pH, and radiation environments. And the spores can be viable for theoretically several thousands of years. So the panspermia option, it may be true that life might have started on Mars and may have been like lifted up in a rock and come to Earth, but it still doesn't solve the problem of how life would have actually started on Mars, unless perhaps the DNA came from somewhere else outside the solar system, but again, that still doesn't solve, where we came from. So considering the origin of life, I'm just going to assume the two main theories. The first is the peptide world. So peptides are chains of amino acids, which are held together by peptide bonds, and these are what fold together to make enzymes. So in the peptide world, this assumes, obviously, the peptides came first. So this is feasible prebiotically. This has been done many times, and we even found a few dipeptides and tripeptides in experiments that simulate the conditions of interstellar space. As I think dipeptides were found in the Murchison meteorite. So this is fairly easy to make. However, they cannot store genetic information or pass it down to the next generation. So therefore, a lot of people think, well, this isn't really life yet. The second one is the RNA world, and this is possibly the most famous. So this is postulated after they found some ribozymes, which are DNA, or RNA rather, that actually folds up and has some catalytic activity, rather like an enzyme. So it's the RNA equivalent of an enzyme. And this is great because it has now the ability to both store information genetically to pass down to the next generation, and also it can catalyze reactions. So theoretically, this, I don't think job's been solved here. However, these are extremely complex. I have on the right here a animation showing a polymerase molecule, which actually has two subunits. These are over 1,000 peptides each. And what's happening is other ribozymes are bringing in, this is called a transfer RNA, is on one end it's reading off the RNA chain, and it's bringing in a peptide on top of there, and that's making a peptide at the end, and another molecule is bringing that away. And you can just see this whole process is incredibly complicated, and these ribozymes are actually extensively modified by peptides, particularly the tRNA molecules. So it's thought that there must be a precursor before this would have been made. So another thing, just very briefly, is that a lot of the theories on the origin of life assume you have a warm little pond. This is because a lot of prebaric chemistries, in particular making peptides, and also some of the parts making RNA, the nucleotides, they are condensation reactions. And therefore, if you have a lot of water, this is a bad thing. So they get around this by basically saying that if you have a warm little pond, perhaps on the side of a volcano, somewhere like this, so you have a volcano here, you have an ocean, and this will fill up with water, but the volcano is still pretty hot. So over time, this will evaporate, and you end up cycling, and you are able to make longer and longer peptides over time in this manner. The alternative is that oceans may be delivered exogenously. And this would help, for example, peptide formation. This is a graph from Lambert et al. 2009 showing between 100 and 200 Celsius, there's a region where peptide bond formation becomes extremely efficient. And it's also possible that this is another calculation of the percentage of the Earth's surface that would have been at a certain temperature during the late heavy bombardment period by Abramov and Moses, 2009. That actually shows a significant amount of the Earth's surface would have been viable to survive, and actually a lot of it would have been in this region where peptide bond formation would have been very efficient. So that's, I think, all I really wanted to say on that. Again, I'll try to give my contact details to Carl, so you can contact me if you have any questions or suggestions. But thank you very much for your time.